Good afternoon, everyone, and welcome to the Health Research Board Trial Methodology Research Network webinar series. Today, our host is going to be Dr. Erika Daly, and she is going to be giving a webinar on randomization in clinical trials, guiding principles and practical applications. To give you a bit of a background on Erika, Erika is the Director of Biostatistics at ICON plc, a top 5 global CRO in the pharmaceutical industry, based at ICON's global headquarters in Dublin, Ireland. She joined ICON as a statistician in 2004 and has supported clinical trials in many therapeutic areas, including oncology, CNS, GI disorders, and cardiovascular diseases. Initially a project statistician, she has been in management roles of increasing responsibility since 2008. As a statistical consultant, she has represented pharma companies in their discussions with the FDA and has served as the unblinded statistician for numerous data monitoring committees. She also has had oversight for the dedicated randomization team within ICON for the past 2 years. Dr. Daly obtained her BA in Statistics and Economics, her Master's in Statistics, and her PhD in Biostatistics from UCD. She is a chartered statistician with the Royal Statistical Society, a former member of the PSI, Statisticians in the Pharmaceutical Industry Scientific Committee, and currently sits on the PSI Regulatory Affairs Committee. So that's quite the CV there, Erika. Before I hand over to you, just for attendees that wish to ask a question about the presentation, please select the Q&A section on the bottom menu of Zoom and type your question in here. You can type your question in during the presentation, and I will ask the first questions for you once the webinar has concluded, time dependent, of course. So with that, I'm going to hand over to Dr. Erika Daly.
Thanks very much, Tom.
No worries.
So today I'm going to start with an overview of randomization and why we do it, and then look at some specific examples of challenges within randomization space. So why do we randomize? Randomization allows unbiased allocation of subjects to treatment by ensuring that each subject has an equal chance of being placed in each treatment arm, whether study drug or control, avoiding the bias in the allocation of subjects to treatment arms. The randomization procedures, i.e. the methods of randomization chosen, should maximize the statistical power, especially in subgroup analyses, by maintaining the desired treatment allocation ratio. Generally speaking, equal group sizes maximize statistical power. However, unequal group sizes may be more powerful for some analyses. For example, multiple comparisons of placebo versus several doses using Dunnett's procedure, and sometimes they're desired for non-analytical reasons. For example, patients may be more motivated to enroll if there's a higher chance of getting the test treatment, or regulatory agencies may require a minimum number of patients exposed to the treatment. We also need to minimize the selection bias, because this may occur if investigators can consciously or unconsciously preferentially enroll patients between treatment arms. So details of randomization that could facilitate predictability should not be mentioned in the protocol. Randomization should also minimize the allocation bias or confounding, which may occur when covariates that affect the outcome are not equally distributed between the treatment groups, and the treatment effect is confounded with the effect of the covariates, i.e. an accidental bias. Random assignments of participants helps ensure that any differences between and within the groups are not systematic at the outset of the trial. Thus, any differences between groups recorded at the end of the experiment can be more confidently attributed to the experimental procedures or the treatment. However, having said that, randomization assignment does not guarantee the groups are going to be matched or equivalent. They may still differ on some pre-existing attribute due to chance, but the use of random assignment, while it cannot eliminate this possibility, greatly reduces it. So what is a randomization schedule? This is the list of numbers assigning what treatment each should be handed out to each person. It needs to be reproducible and secure. Use a stable programming language like SAS to create it. You need to know the seed so that you can reproduce it, but that seed should be kept secret because obviously it would unblind the study if anyone else got access to it. The keeping a paper copy can be important, especially for longer duration trials. We had a situation a few years ago now where there was a flurry of activity on the day we were meant to unblind the study because we realized the randomization codes were saved on a 3.5-inch floppy disk, which of course no one had a computer drive to read, and left us trying to scramble to find a way to access the randomization codes. Another important aspect of the randomization schedule is make it bigger than the study design says you need. At least 3 times as many numbers as numbers of subjects are going to be enrolled allows for larger dropout rates than expected, or a large number of partially used blocks leading to randomization numbers that can't be assigned. There's no extra cost to do this if you do it up front, but trying to extend lists later can become really both technically problematic and costly. And if there's a stratification without limits or targets for enrollment into each stratum, make sure that each stratum has enough randomization numbers to meet these criteria if you assumed that everyone enrolled in the study ends up in just one stratum. We'll talk more about stratification in a moment, but that's some important principles to think about when specifying how your schedule will be designed. So what should a schedule contain? A randomization number, what treatment allocation that number corresponds to, so some sort of treatment code, AB12 or whatever that coding you want to use, and a decode, active, placebo, treatment, blah, blah, 50 milligrams, etc. However that would work. A block number, stratification description, which can either be separate for each stratum or each stratification variable, or one combined stratification that includes all the factors in one variable. And a sequence number is sometimes included for logistical reasons so that the list doesn't have to be sorted by the randomization number. What the schedule should not contain is which subject receives which randomization number. This linkage happens during the course of the study, but the randomization list itself is generated before the first subject is enrolled. So it's important to realize that that linkage comes later. In a simple randomization, it's just a list of random numbers, a corresponding treatment code, A or B, and D code, and a treatment allocation is balanced across the full list. And you can see an example on the right there. However, there may be an imbalance in a subset of the list. For example, in a list of 100 subjects, 50 will be assigned treatment A and 50 will be assigned treatment B. But it does not follow that the first 10 subjects on the list, there will be 5 A's and 5 B's. In fact, it could look like this example here, where in the first 10 subjects, there's only 1 A and 9 B's. This can be very problematic if it's expected that there is a difference between early and late enrollers in the study. For example, early enrollers may be the more severe cases who agree to participate immediately the trial opens, whereas later enrollers may be milder cases. Or it could be because of different centres or even different countries enrolling at different times. For example, Poland takes about 6 months to open a site compared with 4 months in most other European countries. China and parts of Latin America can be up to 9 months to start, but would then be high enrollers afterwards. And this could also lead to confounding. It can also be a problem if DSMBs or interim analyses are planned for regular unblinded monitoring during the trial, or if the trial is stopped early for any reason, as you may not have sufficient numbers in each treatment group in order to perform a comparison if you've had unequal allocation up to that. A way around this is block randomization. Blocked randomization ensures the balance is maintained in each of the subsets. Balance is ensured by allocating at random, but with the constraint that allocation within the blocks occurs in the correct proportions. Excuse me. For example, equal numbers of A's and B's for a 1-to-1 randomization ratio. In a blocked randomization, though, the choice of block size or length, i.e. the number of allocations within each block, is very important. It depends on the number of treatment arms, and should be a multiple of that, but too big or too long, and the same issues with imbalance that were in the simple randomization will occur. However, if it's too small or short, it means the allocation could be predictable. So if there are 2 treatments and block size is 2, then you know that subject 1 and subject 2 are on different treatments. You may not know which is which, but it is still a partial unblinding that could lead to those subjects having to be withdrawn from the study. Generally speaking, it's recommended that 2 to 3 times the number of treatment arms is used as the block size, but with the proviso that this is a practical size. So for a dose-finding study with 6 treatment arms, that would imply a block size of 18, which is likely to be far too large and lead to the same imbalance problems of the simple randomization list. The total number of blocks needed is simply the total number of randomization numbers needed divided by the block size. So if you want 100 subjects on your randomization list and your block size is 4, then you need 25 blocks. If your block size is 6, then you would need to increase the list to include 102 subjects and you would have 17 blocks. So here's an example of a randomization schedule with 2 treatment arms in a 1-to-1 ratio and a block size of 4. So you can see in the first block, 2 A's and 2 B's, 2 A's and 2 B's in the second block, 2 A's and 2 B's in the third block, etc. They're in different orders each time, but it allows that if you take, for example, 8 subjects, you will have an equal randomization allocation as per the design. The next type of randomization we're going to discuss is stratified randomization. This is where we stratify subjects by important prognostic factors at baseline, for example, age, sex, disease severity, because this may sometimes be valuable in order to promote balanced allocation within stratum, which has a greater potential benefit in small trials. The levels of known factors or a combination of factors can then be included in the randomization design. Stratification by more than 2 or 3 factors is rarely necessarily and is generally less successful at achieving balance and is logistically troublesome. So where possible, restrict your stratification to under 3 factors. All stratification factors included in the randomization design must be accounted for in the subsequent analysis. So that is also something that needs to be factored into your design. So here is an example of a stratification analysis. Stratum 1 is males with moderate disease severity at baseline, and there are 100 randomization numbers in this stratum. Stratum 2 is also males, but with severe baseline disease severity, continuing into stratum 3 for females, moderate and severe, and 100 randomization numbers within each stratum, 25 blocks, and the corresponding treatment codes. A recommendation I have is that the randomization number for each stratum begins with the number of that stratum. It's not necessary because you set the range for the randomization numbers in this randomization design, but logistically it is helpful to have a direct link between the randomization number and the stratum. Strata are not blinded, nor do they affect predictability of treatment allocation because they are based on the known characteristics of each subject. Thus, this doesn't affect prediction of the treatment codes. But it also allows that if there are additional randomization numbers to be inserted, if you need to extend the list in future, that they can be inserted with the same link to the stratum without affecting randomization numbers already assigned. So if we needed, for example, another 100 moderate males to be added in, they could be 1101 to 1200, and that would have no impact on the male severe group or on any of the other strata. There's one more type of randomization I'll talk about today, which is dynamic randomization, also known as adaptive randomization. This uses algorithms to assess values for subjects already randomized and to allocate the next subject to the treatment arm that minimizes the current imbalance in those values across treatment arms. Values assessed are either based on the covariate profile of existing subjects or on the responses they have shown to the treatment they have received. One issue with stratified randomization is that if there are a lot of factors, the number of strata increases rapidly to the point where sample size to ensure sufficient numbers of subjects in each stratum would be prohibitive. Minimization looks at all the specified covariates collectively assesses the degree of imbalance in each of these covariates that would be created by assigning the subject to each of the treatment arms, then sums them to give the overall imbalance in the study. The algorithm then selects the treatment arm that yields the smallest imbalance between the groups overall. This method can be useful when there are a large number of factors thought to influence treatment response, and there is the expectation that simple randomization will not adequately maintain the balance of these factors at baseline. The other main form of dynamic randomization reviews the responses to treatment, either the main endpoint or a surrogate, of existing subjects, and adjusts the probabilities associated with each treatment arm. So the probability of being assigned to a treatment arm increases if the responses of the prior patients in that arm were favourable. This can be useful when trying to model a dose-response curve, where many possible doses are being assessed simultaneously, by putting fewer subjects in the ineffective or non-tolerated ranges. However, this method requires a response measure or endpoint that is very quick to obtain compared to the rate of recruitment. Otherwise, there will be no response data for the algorithm to use to adjust the probabilities. PK data, such as maximum concentrations, Cmax, or time to maximum concentration, Tmax, can be useful with response-based dynamic randomizations. But ultimately, the success of any dynamic randomization depends on the quality of the algorithm used to decide how the subject's values change the the allocation strategy, and also on the quality of the implementation of this algorithm. So a great algorithm, badly coded, is useless. The complexity of the logistics and potential impact on the analysis should be carefully evaluated when considering whether dynamic allocation is a viable option for your study design. I'd like to talk about another aspect related to randomization, that is balancing. ICHE9, the International Consortium on Harmonization Statistical Principles for Clinical Trials, recommends stratifying by centre, or equivalently assigning full blocks to centres, which is balancing by site. This is logistically simple, as it effectively divides the randomization list into distinct parts, with each centre receiving one of those parts and thus able to randomise independently of other centres. To illustrate this, taking the block randomization schedule from earlier, assume that the first 5 subjects into the trial are given by these numbers. So S001 at site X, so X01 is the first subject at site X, the first subject at site Y, a second subject at site Y, a third site opened with 1 subject, and finally a second subject at the first site. This is how that would look. So here's our blocked randomization schedule. The first subject comes in and so site X gets block 1. The next subject is at a new site, so goes to site Y, gets block 2, and then subject 3 is also at Y, so it's in the same block. Subject 4 is at a new site, Z, so goes to a new block. Subject 5 is back in the first site and back to the original block. So you can see they jump back and forth as they enroll. But if you look at the corresponding treatment codes, you can see that in the first 5 subjects, there are 4 A's and only 1 B. When there are a large number of sites with few subjects recruited at each site, incompletely filled blocks can lead to imbalances overall. There are also gaps in the assignment list. So for example, randomization number 1003 is not assigned to anyone, even though higher randomization numbers are used. Technological advances in the past 15 years mean that balancing across centres is now feasible. This is also known as balancing by study or centrally balancing. In this case, subjects are assigned the next available randomization number and associated treatment arm, regardless of what site they are at. This requires a system that can centrally monitor randomization at all times and ensure that the correct randomization number is assigned each time. It also impacts how treatment kits are labelled and distributed to subjects at each site. So in this case, the first 4 subjects are just the first block. The 5th subject is in a new block, and that is independent of the site. It is just in the order in which they arrive. Some logistical challenges to consider in relation to this is that stratification by site can mean many unused treatment kits at the end of the trial, but transport costs or time may not allow for just-in-time resupply. So you need to be aware of the practical constraints of the method chosen as well. Creating a separate list for labelling treatment kits, a pack list, can offer more flexibility in drug allocation. Kits are not pre-assigned to a randomization number or subject, so unused kits can be reassigned if there are dropouts. When treatment arms are made from combinations of dose levels, kits can be matched as needed. So 2 actives is the high dose, 1 active, 1 placebo is the medium dose, 2 placebo is the low or 0 dose. And for multi-center trials, the unused kits at one site may be able to be returned to a central depot and redistributed to other sites. For example, if a site was closed for non-recruitment, which enhances the flow of medication you have available, which is particularly useful in trials where there is low drug availability. So what is a pack list? It's created the same way as a randomization schedule and subject to the same security constraints we've already discussed. The schedule needs to contain a kit number or pack number, whatever you wish to call it, the treatment allocation, the code and decode. There's no need for stratification or blocks because they only apply to the randomization of subjects, not to the labelling of the kits. The kit numbers allocated are recorded on the drug dispensing log or exposure page in the clinical case report forms, the records of the trial. Typically, much more kit numbers are required than randomization numbers. So for example, in a situation where the dosing schedule requires 1 bottle of tablets at visits 0, 4, 8 and 12 for a 16-week trial for 120 subjects in a 1-to-1 ratio. Well, 4 kits per person times 120 subjects is 480 kits needed in the trial. Multiplying by our overage of 3 means that we need a minimum of 1,444 kit numbers, whereas only 360 randomization numbers. The 120 subjects by our multiplier of 3 would be needed on the randomization list. Ideally, use completely different numbers for kit numbers and randomization numbers to avoid confusion. The last thing you need is for someone to get a randomization number and give the person the kit that matches that randomization number instead of checking what kit that person should be assigned. For an example, 5 digits beginning with 8 for kits and 4 digits beginning with 1 for randomization numbers, subject numbers, means that you can't get confused between which numbers you're talking about. A central principle of statistics is that the analysis must follow the design. If you stratified your randomization, then all stratification factors must be included in the analysis. So for example, you want to adjust for continuous covariate in the analysis, like age, then you cannot stratify by age categories over or under 65 years of age in the randomization, as that dichotomized age would have to be included in the model, but dichotomized age and continuous age would obviously be correlated. And therefore you wouldn't have the option of including continuous age in your analysis. Of course, you can include covariates in the analysis models that were not part of the randomization design, so it's worth considering whether the stratification at randomization is really necessary or if adjusting for these factors at the analysis stage might be sufficient. At unblinding, the randomization list, the subject list, and the pack list are merged together with the clinical database to determine the treatment allocation. You merge by the randomization number to get the planned treatment. You merge by the kit number to get the actual treatment they received. And then you must have pre-specified rules about how misallocations will be handled in the analysis. So if somebody was planned to get placebo, but actually received the active dose, how are you going to analyze them? As planned or as treated? Creating dummy lists to practice the unblinding procedure and create blinded results during the course of the trial can be very helpful. Then when you get to the final unblinding stage, only the list needs to be changed, but the programming is already in place, which allows for quick delivery of unblinded results once the trial ends. If the blind needs to be broken during the trial, for example, for safety concerns, you need to have 24-hour access, especially in global trials, to those trial codes. So who holds these codes for a multi-center trial? Does the code holder have the necessary languages to support a multinational trial? If you have a central randomization system, IVRS or IWRS, which is interactive voice or web response system, then this can be used to unblind individual subjects. Code break envelopes are a paper-based option, which can either be held at each site or in a central location that would be contacted. But the full list should not be used as a source for individual unblinding because of the risk that while looking up one subject, you see and unblind several others at the same time. I'm now going to look at some specific examples of more challenging randomization designs and how to work around them. The first is sentinel dosing. Some of you may recall the theralizumab at Northwick Park disaster in 2006, where in a phase 1 clinical trial, suddenly huge numbers of pretty much all of the patients who'd been given the active drug came out in very serious complications, side effects, and many were hospitalized and permanently crippled from it. As a result, new rules were released, the guideline on strategies to identify and mitigate risks for first-in-human and early clinical trials with investigational medicinal products. The European Medicines Agency, the EMA, finalised this guideline this year. Section 8.2.6 of that states that when the study design includes the use of placebo, it would be appropriate to allow for one subject on active and one on placebo to be dosed simultaneously prior to dosing the remaining subjects in the cohort. This pair of subjects, the first people to receive the drug, are known as the sentinel pair. Usually 24 hours is allowed between dosing the sentinel pair and dosing the rest of the cohort to give time for any adverse reactions to make themselves known. So an example of this, you have a cohort of size 8 in a 3-to-1 ratio overall. So in total, you want to have 6 active and 2 placebo subjects. In a 3-to-1 ratio, you might have a block size of 4, so you'll have 2 blocks to make the whole cohort. In sentinel dosing, the first 2 subjects are known to be 1 active and 1 placebo, kind of a 1-to-1 ratio, and the remainder of the first block therefore contains 2 active. So a standard block design would mean the unblinding of a quarter of your subjects, because you would be absolutely certain what subject 3 and subject 4 received in your trial. Obviously, this is not desirable in any sense, so the way around this, in terms of randomization design, is that if sentinel dosing is required as part of your study design, use unequal blocks. Block 1 will have size 2 and a ratio of 1-to-1 for the sentinel pair. Block 2 will have size 6 in a ratio of 5-to-1 for all the remaining subjects. The next example is where there is a need for re-randomization during the course of the trial. So, for example, a study design where there's an initial randomization 1-to-1 to placebo or active for a 16-week efficacy assessment, double-blind. And then after that, there's a 24-week follow-up period for safety. Active studies remain on the same treatment during follow-up, i.e. they stay on active throughout, but placebo subjects are re-randomized. But how do you implement this without breaking the blind for the efficacy assessment if only half of your subjects need to go through that re-randomization process? There are 2 standard options for what happens to the placebo subjects. Option 1, everyone is redirected to the active, or option 2, some are redirected to active and the rest remain in placebo. We're going to look at each of those options now. So in option 1, all placebo subjects are re-randomized to active. Well, the way to stop that breaking the blind is we don't re-randomize. Instead, after the 16-week efficacy period, move everyone to to a new common treatment arm called the open label active. So by acknowledging that everyone will be on active regardless of what they started on, you can avoid that sense of having to say that only placebo subjects need to go through this additional step, and thus don't have to reveal who the placebo subjects were. In the case of option 2, where placebo subjects are re-randomized 1-to-1 between active and placebo, in this case, don't randomize 1-to-1 to treatment arms initially. Instead, randomize 1-to-1 to 2 to treatment sequences. So a quarter of your subjects start on placebo and move to active, a quarter start on placebo and remain on placebo, and half of your subjects remain on active throughout the trial. This may not be possible, for example, if there's a high dropout rate expected, so in which case you would have an unequal balance at the end. If you do need to go through a re-randomization step, make it a stratified re-randomization step. So the initial treatment arm tells you which outcome you have. If you were placebo, in that stratum you can either go to active or placebo. If you are active, you only have one choice, you go to active. But all subjects go through the re-randomization step. Thus, you don't reveal which subjects were the placebo subjects by having to do the re-randomization. My final case study of challenges with randomization design is how to maintain the treatment ratio in small studies where more than one site is involved. So, having again a cohort of size 8 in a 3-to-1 ratio overall. In this example, there were no blocks. We just only 8 subjects, a simple randomization list, 6 active and 2 placebo subjects. But then a second site was opened. So there are 2 sites, each of them gets 1 block. And assuming they recruit evenly, you could have the situation that the 4 subjects from site 1 and the 4 subjects from site 2 are all active subjects, because the 2 placebo subjects in both groups could have been in the last 4 of their block of 8. Or, in the reverse, you could have 2 placebo subjects at each site for an overall ratio of 4 placebo and 4 active, a 1-to-1 ratio instead of a 3-to-1 ratio overall. And you could have any combination in between, and that's assuming that they recruit evenly. One way around this is to change the block size to 4. So therefore you know that you'll have 3-to-1 in the first 4 subjects and 3-to-1 in the second 4 subjects. This does lead to a risk of unblinding a whole site if the 1 placebo subject happens to be unblinded, because then you also know what the other 3 subjects at that site have received. If one site recruits more than the other, the ratio of 3-to-1 may not be preserved also, for the same reasons as above, because you don't know what the first number of the next block is going to be. The best conclusion is that if you're using multiple sites, use an IRT platform, an interactive response technology platform, to manage your randomizations, rather than trying to do this manually through paper approach. In summary, keep it simple. Minimize the number of stratification factors included in the randomization. Only use dynamic algorithms if there is a clear benefit or need for this given your study design and objectives. Be aware of the practical constraints as well as the scientific objectives. Decide whether you need to balance centrally or by site. Do you have the infrastructure available for central balancing? And is balancing by site suitable for your trial design? Remember that the analysis must follow the design, and consider this when choosing stratification factors and other randomization design elements. And for anything more complicated, I would always advise that you consult a statistician and get advice on this, because if the randomization is wrong, everything else that follows becomes meaningless and worthless. So you need to make sure that this critical step is right. Thank you very much.
Thank you very much, Erika. That was a very interesting and useful talk. So if there's any questions, people can type them in to either the Q&A box or the chat box. And we have one question already from Declan, and he's wondering, in your view, when is a participant recruited to a trial? Is it the point of invitation? Is it screening? Is it consent or randomization? Because he thinks this doesn't seem to be reported clearly in trials, and his own view is that recruitment happens at randomization, but he's aware that not all will agree.
My experience from clinical trials is that there are two numbers tracked: the number enrolled or screened and the number randomized. The number randomized is the number that actually matters in terms of the analysis. But if there's a high screen failure rate, and usually from a practical point of view, that's also something that is being tracked. So you might be looking to screen 100 subjects in order to randomize 40 of them, if there was a very high screen failure rate, for example.
Yeah.
My experience has always been that both of those figures are tracked and reported, and that in terms of timelines for trials, the first subject enrolled, which is screened, informed consent signed, and then initial screening performed, is a date that is tracked, as well as the first subject randomized date.
Thank you very much for that.
Okay.
So we have another question. Are examples of embedded factorial designs in trials common? For example, if the intervention arm is allocated X and then we randomize that group to A via B. I think does that make sense?
No, can you say that again, please?
Are examples of embedded factorial designs in trials common? In the example here is if the intervention arm is allocated X and then we randomize that group to A VB. I'm not sure what it means by VB in the middle.
Versus B. That would be
A versus B. My fault.
Yeah. I'm not sure I understand your question, Declan. If you're allocated them to one treatment arm, then how could you also allocate them to a comparison? You will have multiple treatment arms. Who you choose to compare between those treatment arms is part of the study design and objectives. It's not really related to the randomization itself. It's more the analysis subsequently. Have I misunderstood?
What we'll do is, if there's a couple of more questions coming up, so if he wants to retype and see if it's coming across the way he wants it to come across.
Okay.
We have a question from Rustam, and he says, can you give some examples of the worst randomization mistakes that you've encountered?
I can see what the actual word is there.
Okay. We won't say it out loud.
Okay. Yes, so one of my favorite ones is Stephen Sen, which he described as, rather than incomplete block design, was incompetent block design, where he had a very detailed randomization schedule and each site had to recruit in a particular order in order to have this beautiful overall balanced design. But what actually happened was the site said, I'll just go to my fridge and hand out what I've got. Completely destroyed the design and all of the principles that allowed a very small sample size to be used because of the power of the randomization design was lost because an investigator decided that what was in the fridge was more appropriate, or that he knew better than what the randomization scheme said. I have a similar example of that, whereby, the site, it was a dual system where there was an IRT platform being used. The site received a randomization number and was told, therefore, you need to allocate, from the randomization number, the system knew that this subject was going to be assigned to treatment A, and looked on the shelf, the dispensary of the site, to say which kit number should be handed out to be treatment A. And the response was, cannot be randomized at this time. That's code for, actually, you have no treatment A left on your shelves. We need to get you a resupply. But the investigators decided, well, I've got the family sitting here right now. I'm not going to ask them to come back later. And I've got other people lined up to randomize today. I'll just give them what I have. And the next 4 patients were all given stock from his shelf, which, of course, to those of us who understood what had happened, meant that we knew that every single one of those 4 subjects was on treatment B, and thus were partially unblinded to a chunk of the trial because we knew that he only had treatment B left on his shelf because he hadn't waited for the resupply. So those are some of the examples, and maybe it's unfair of me to highlight the ones where investigators have overruled the randomization design. That usually is the biggest challenge. It's not so much the design as not sticking to it once it's being created.
Okay, thank you for that, Erika. We've got another question here, and it's from Marie Galligan, and she says, thank you, Erika, for the interesting talk. I'm working on a multi-site study with around 10 sites. Variable block randomization is used, stratified by site. As you mentioned, if sites do not complete blocks, this can lead to imbalance in treatment arm size. We didn't plan to use an IRT system, but will in the future. In this case, could we specify for each site in advance the number of patients that they should attempt to enroll?
Yes, you could give each site a target. In my example of maintain the ratio in small studies with more than one site, what we actually did in that situation was we got 2 sites and we told them that they each have to recruit exactly 4 subjects, half the cohort. And even if one site has an extra subject to recruit and the other one is lagging behind, that's not an option. It's 4 and 4 because anything else would affect the ratio. In a very large trial where you have lots and lots of centres, incomplete blocks don't have as much of an impact because some you'll be short on A and some you'll be short on B, and on average, they'll cancel out. But when you have a very small number of subjects, even 1 or 2 in the difference can have a huge impact on the overall ratio. So yes, you can set target numbers per site, but you need to consider how that fits in terms of your block size and also how that fits in terms of your timelines, because presumably you opened multiple sites with a view to recruiting more quickly. And if one site is failing to recruit their target, you need to then decide how long are you willing to wait for one site to finish that block versus allowing other sites to recruit patients they found available already.
And Marie has written to say thank you. That's very helpful. Okay. So Declan has made his question a bit clear. So the original question was, are examples of embedded factorial designs in trials common? So if, for example, a participant is randomized to an intervention to determine effectiveness of the intervention against control, but then that intervention group is randomized a second time to, for example, different levels, intensity of the intervention. Seems like an embedded factorial design, but perhaps not. What would you think?
I think this comes down to similar to my re-randomization example, whereby after being randomized to an initial group, you're then randomized to a different group again. And yes, I think in that case, Declan, when you have the group who were on placebo during the efficacy period and are now half are on placebo still and half have moved to active, you could compare within that group for those who've had an initial period, a lead-in period of placebo, now what the change is. You could also compare the person who's on active in the safety period with the person who was on active throughout the whole period to see whether that initial 16 weeks has a different impact then, or if it's the more recent medication history that is more telling to the outcome. So yes, there are different analysis strategies you can do, and personally I hate it when study designs call for any sort of crossover treatment and switching of arms, because it muddies the water as to what your comparisons are actually showing and what you can draw as an interpretation from the study thereafter. A clean parallel group two-way comparison is always much simpler, because what you need to consider is not just maybe a really novel design that can minimize the number of patients and can be really statistically efficient. What message will you have at the end of it? A really simple message that you can bring to regulators, doctors, patients, and say, we did this test and this is how you know that this drug is going to be good for you. If your design is so convoluted that people can't even work out what question you asked, then you're going to have a difficulty explaining that the results you got do actually justify the question you set out to answer.
That's great. Declan says thanks a lot. Good point. That's clear. We have one more question here. and it's from Dennis Shields. And he says, do the principles outlined here to blinded studies apply equally to unblinded, or does that design give greater freedom?
Absolutely. Whether a study is blinded or unblinded is actually a separate issue from whether it's randomized or not. Normally we talk about randomized double-blind trials and the terms become almost synonymous, but the opposite of randomized is observational, whereby we just see who comes in and see what happens to them. So the principles of randomization, minimizing bias, removing confounding factors, etc., apply regardless of whether the subsequent observation or the treatment is revealed to the subject or not. There are some things that are maybe logistically simpler in that you don't need as much, maybe you don't need a pack list, because you can just say, right, this subject is going to be on the active treatment group, just prepare the active medication and hand it to them, as opposed to saying, give them kit number 10351. So there's maybe a little bit less logistical complexity there, but in terms of the statistical principles, whether or not the treatment is revealed to the investigators, the analytical team, or to the patient has no bearing on the principles of the randomization design.
That is great. Thank you very much. And I think with that, we will, Dennis writes in to say, thanks, that makes sense. We have one more question if you have time, Erika, I'm not sure. There's a question saying, I'm guessing that the randomization concepts mentioned earlier can be applied in adaptive trial designs.
Oh yes, absolutely. So, but adaptive trial design is an umbrella term for an awful lot of different things. So to clarify on what that means, if we're talking about adaptive in the sense of the sample size can be adjusted, or potentially a future treatment arm could be dropped. So let's start with sample size can be adjusted. Yes, the randomization can be changed. The randomization design will not be affected by that. It'll just keep going on for longer. An important point is to note what is the maximum sample size you could be doing and create your randomization list to be large enough for that maximum possible sample size rather than the sample size you start with, so that you don't run out of randomization numbers halfway through. Another type of adaptive design is where you're choosing treatment arms. So you maybe start with 3 active doses and a placebo, and you're doing an interim analysis, and after that interim analysis you're going to pick 1 dose level to continue and the placebo arm. So then the question is, how is that study designed? If anyone who isn't on the dose level that is being continued is just going to be discontinued from the trial, then it's just an arm has been stopped after that point, so it doesn't affect the randomization. If there are new people going to be recruited, you'll need to have a new randomization list that is now in a 1-to-1 treatment ratio with, chosen dose, unspecified in the initial scheme, which can be then entered in after the interim analysis. And your original list would have 4 doses, 3 active and placebo for that allocation. So it depends on what your study design is and how the adaptation is going to impact it. The randomization can reflect that in many ways. But if you're going to be doing an adaptive randomization as opposed to an adaptation of the study design, where you have no idea what doses you're going to pick and you're going to decide based on what you see, then you're very much in the dynamic randomization space, and need to consider very carefully how you will structure that randomization list to give you maximum flexibility for possible arms that will open or not. What I have seen done in the past, especially where you have an IRT platform available to you, is that you create cohort and you label them. So this is now cohort 10 is going to be called cohort X, MG. Then cohort 11 is cohort Y, MG. Cohort 12 is cohort Z, MG. Because what that is you can specify the ratios for cohorts X, Y, and Z, even though you don't know what dose of treatment will be assigned to those arms, treatment arms within the study. Then your IRT system usually can relabel that treatment description, so that when it comes to the point that the data in the trial suggests, yes, we will open a new dose arm. We have a 25 milligram dose and we have a 30 milligram dose. Now we're going to open a 40 milligram dose. You can just relabel that X milligram dose to be 40 milligrams, but you've already got your randomization allocations set up, ready to open that treatment arm and just start recruiting into it. So there are ways you can use the technology you have to make that easier. And what I would advise when designing a randomization is know what platform you're going to be using, whether it's paper-based or whether it is some IRT platform. Know what its strengths are, know what its limitations are, know what's possible, because there may be easier ways to implement the randomization design if you can leverage some of that technology. There may be things that you want to do in your randomization design that just aren't possible by the technology you're using, and thus you need to have a different design. So it's important that you consider those logistical and practical challenges of how would we actually make this happen at site, as well as what is the optimal design in a theoretical concept only.
Well, Erika, that's how you answer a question properly anyways. That was, thank you to people who asked the questions. But a huge thank you to Dr. Erika Daly for the time and effort she put into this presentation and the detail that is evident to hear. I personally found it very useful and informative, and I hope everyone who attended did also. So thank you very much for your time, and just to let everyone know as well that this webinar will be available on the HRB-TMRN website, hopefully by either this evening or by tomorrow. And we have a few thank yous in from Declan Devan and from Catherine Gorecki also. So a very enjoyable talk. And yeah, again, thank you very much, Erika.
Thank you all very much for having me. It's been a pleasure working with you today.
Excellent. Have a good evening. Have a good evening, everybody. Bye-bye.
Thank you. Bye.